In this presentation, we will discuss the benthic invertebrate lab methods used in our Farmington Bay wetlands research in 2020. Research was conducted by Oreohelix Ecological and members of the Wasatch Front Water Quality Council. Goals of this portion of our study were to 1. Estimate benthic invertebrate densities and dry weight biomass in Farmington Bay wetlands throughout 2020. 2. Measure the effects of bird predation on benthic invertebrates. 3. Focus on major invertebrate taxa with low resolution taxonomy, mostly to family taxonomic resolution. For our field processing methods, see our YouTube video at this link. We use 3 types of magnification: an illuminated magnifying lens, a dissecting microscope, and a compound microscope. We used a drying oven and an electronic balance to weigh the dried macroinvertebrates. Live invertebrates were hand-picked from detritus within 48 hours after collection from the Farmington Bay wetlands. Typical picking times were between 4 and 8 hours per picker. Residuals from primary picking were then quality control checked by a second different picker. Residuals were then combined with original pick samples. In these images, clean, hand-picked samples are ready for taxonomic separation, counting, and oven drying, and dry biomass weights. Taxa were identified using Merritt and Cummins. Midge, or chironomid larvae, were identified to Chironomini, Tanitarsini, Orthocladinae, or Taniponidae. Midge larvae were by far the most abundant benthic taxon in the wetlands. Midges are in the family Chironomidae and are often called chironomids. Larval taxonomy below family level is typically only taken down to subfamily or tribe. However, experts can identify midge larvae to genus and on occasion species. Larval mouthparts are required for taxonomic ID. In Figure 1, the striated ventrometral plate distinguishes the tribe Chironomini, of which the genus Chironomus are a member. The mouthpart, known as the ligula, shown in Figure 2, distinguishes the tribe Tanitarsini, of which Tanipus neopunctipedis is a member. The red midge larvae shown in Figure 3 are Chironomus, collected from a 0.1 square meter sample from the wetlands in May 2020. Whenever we had taxonomic questions or concerns, we summoned Brett the Bug Guy and his minions at River Continuum Concepts. Processed benthic invertebrate samples were placed on pre-weighed aluminum foil trays and then dried at 100 degrees C for 4 hours in a drying oven. Dried samples were then weighed to the nearest milligram on an electronic balance like the one shown in this slide. For related videos, check out these YouTube links.